team includes Vũ Phương Linh, Nguyễn Hoàng Dương, Nguyễn Lê Hà Chi, and Đặng Lâm Yến Nhi. Today, our team is going to talk about Anthraquinone from Vietnamese Morinda citrifolia L, alleviate good pain via NLRP3, inflammation inhibition. As you know, GERD is a type of arthritis, which is a group of related disorders caused by episodes of abnormal inflammation in the joints. People with GERD have high levels of a substance called urate in the blood, hyperuricemia. When you have GERD, your body either makes too much uric acid. When uric acid builds up in the fluid around the joints, tiny crystals called urate crystals can form. The activation of NLRP3 inflammation requires two signals. The first, priming signal, is needed for the expression of inflammation components. The first signal may be initiated through pattern recognition receptors, PRR, or cytokine receptors, such as TNF-alpha or IL-1, and induces the signal transduction pathways, mainly including P38, ERK to regulate transcription mechanism. In myeloid cells, ASC and procaspin-1 is highly expressed, and the first signal basically ensures that expressions of NLRP3 and pro-IL-1 beta in a suitable amount. Crystals cause GERD symptoms, including pain and swelling. The second signal is activating signals, which results in the formation of inflammation complex and the subsequent activation of caspin-1. The activated caspin-1 ultimately cleaves pro-IL-1 beta and pro-IL-18, leading to the maturation and secretion of these pro-inflammatory cytokines, depending on the environmental conditions. Morinda citrifolia has attracted considerable attention due to the presence of pharmacological and phytochemically important alkaloids. Especially, anthraquinone has tremendous pharmacological actions, including anti-inflammatory, hepatoprotective, hair growth stimulant, and anti-tumor activity. Our aim in this project is to isolate anthraquinone from Vietnamese Morinda citrifolia L fruit, explore novel functions of anthraquinone from to anti-good pain via the specific inhibition of the NLRP3 inflammation in macrophages. And the last aim is to evaluate effects of anthraquinone on MCSU-induced acute GERD in mice. Next, Dr. Amy will continue our team presentation.
Hello everyone, my name is Đặng Lâm Yến Nhi, so now I'm going to do my presentation. So as you can see here, this figure 1, we show the installation of the anthraquinone from Vietnamese Morinda citrifolia L fruit. Now look at the next is figure 2. We show the TRC and HPLC of the standard and sample anthraquinone. The next one is figure 3. To evaluate the anti-inflammatory of anthraquinone, we examine the cytosine effect of anthraquinone and colchicine on BMDMS. BMDMS were treated concentration of anthraquinone and colchicine, and the percentage of the surviving cells were counted using CCK8. So now look at the figure 4, as you can see here. So now I'm going to talk about figure 4. The figure 4 is the anti-inflammatory priority of anthraquinone concentrations were confirmed by examination of their effects on the pro-inflammatory cytokine production, which is IL-6, TNF-alpha, IL-1 beta. And the result indicate that the release of IL-6, TNF-alpha, and IL-1 beta was strongly inhibited in BMDMS compared to the untreated control cells. And now look at the figure 5, as you can see here. The anthraquinone suppresses MSU indicated ROS production. BMDs were incubated in concentration of anthraquinone for 20 minutes and stimulated by MSU 500 nanometers crucible for 3 hours. CO is collagen and UA is anthraquinone. So now I would like to introduce the next speaker, is Fu-Ling.
Hello, I'm Fu-Ling and this is figure 6. Since the NLRP3 inflammation is responsible for anthraquinone maturation, we then examine the potential effects of anthraquinone on NLRP3 inflammations in the cytoplasm. LPS treatment alone for 12 hours could trigger the expression of NLRP3 protein and brought to kill 1 and retreatment of anthraquinone for 20 minutes suppress the increase of NLRP3 and brought to kill 1 And next is the figure 7. The reduction of MFCU-induced pain was significantly by administration of NLRP3 anthraquinone 40 mg per kilogram. And the last is the figure 8. We further investigated the regulatory role of anthraquinone in the secretion of inflammatory mediators in mice injected with MSU. The serum level of interkilo 1 was significantly suppressed in mice that had administered anthraquinone 40 mg per kilogram. And we come to the conclusion. This study demonstrated that anthraquinone 30 mg per milliliters scavenged oxygen-free radicals, suppressed NLRP3 inflammation activation, and inhibited the production of inflammatory cytokines. Thereby, anthraquinone contributes to the amelioration of gut pain. And the last but not least, anthraquinone 40 mg per kilogram represent potential candidate drugs for the gut pain in the clinic. That's the end of my presentation. Thank you for your listening.